Hi, I'm Larry Johnson. I'm a professor at Texas A&M University, and today we're going to talk about the male reproductive system.
Male reproductive system, the testicular cells, including Leydig cells, Sertoli cells, and germ cells, and the germ cells themselves are the Spermatogonia, spermatocytes, and spermatids, mitotic and meiotic metaphase plates in dividing cells.
Here we see the Leydig cells, and we actually see some lipid in the Leydig cells, and the seminar for tubules. And you can see a host of germ cells, these round ones, these bigger ones, primary spermatocytes, spermatids, and elongated spermatids that you see there. Now, testosterone needs to be higher concentrations in the tubules than the rest of the body, and therefore you have Leydig cells distributed throughout the testis. So in the seminary tubule, we see germ cells of different types. You've got a spermatogonia on the base, spermatocytes above there, and then you've got spermatids. And here we can see the spermatids in through there, spermatocytes over here. And then also we've got a Sertoli cell. Here's a Sertoli cell nucleus here, kind of pear-shaped. You can see them here and here. These are nuclei of the Sertoli cell. So in the tubule itself, you have germ cells and Sertoli cells, and surrounding around the tubule you have myoid cells. So we see fibroblasts throughout the interstitium, which is what's between the tubules. You see the Sertoli cell nucleus, and we see the myoid cell nuclei in through here. A myoid cell marks the limit of the outside of the tubule, and then we have spermatogonia. Sometimes they're dark, sometimes they're light. If we look at this light microscopic view, you can see endothelial cells, there's blood vessels. Anytime you got endocrine function, you're going to have a rich vascular supply. Here we have spermatogonium. There's a Sertoli cell nucleus there, and there's one here, a nice pear-shaped Sertoli cell nucleus. And then we see the spermatids. So these are nuclei spermatids, and we see a little cap, acrosomic vesicle, and a little cap phase spermatids, and these are the ones that look like bullets. These are the ones that mature. So you got spermatogonia, spermatocytes, and spermatids. And we can see those in a drawing and in the real tissue. Here we see the spermatogonia, the pale ones, but there's also dark spermatogonia too, again on the base. And then we can see above the base we have spermatocytes, and we can see they vary in type 2. These are younger than these ones. These are packetine primary spermatocytes. And then there's secondary spermatocytes. You can see the secondary spermatocytes here. And then when they divide, they give you spermatids. And the spermatids come out spherical and look very similar to the secondary, except they're smaller. The chromatin looks very similar, but they're smaller, half the size. And then we get mature spermatids. Mature spermatids as a differentiation of the round spermatids, the young spermatids that we see there. There's also Sertoli cell nuclei. There's one there, one there, one there, one here as well. And there's myoid cell nuclei, as we can see, and the myoid cell marks the limit of the tubule. And so if we look along the base where the spermatogonia are, we can see mitotic figures. So these are mitotic figures in the spermatogonia, as opposed to these guys. These are meiotic figures where you have dividing spermatocytes. So dividing spermatogonia versus dividing spermatocytes. And it has to do with whether they're off the base or whether they're up off the base in terms of location in the height of the seminal epithelium, and because as they move forward, they mature.
We want to acknowledge the original sources of diagrams and some of the images that we're seeing that were taken from textbooks, and they are acknowledged here. So that's the end of the male reproductive system, the testicular cells, including Leydig cells, Sertoli cells, germ cells, mitotic and meiotic metaphase figures in dividing cells.